This video covers the classification and biological importance of lipids. We will explore lipid chemistry, including definition and classification, and their biological importance. Lipids are organic substances relatively insoluble in water, but soluble in organic solvents like chloroform, ether, and benzene. Lipids are classified into three main types: simple lipids, complex lipids, and derived lipids. Simple lipids are esters of fatty acids, or FA, with various alcohols. They are subclassified as neutral fats or oils, where the alcohol is glycerol, and waxes, where the alcohol is other than glycerol. Neutral fats or oils are uncharged esters of fatty acids with the alcohol glycerol. This slide further shows the structure of neutral fats or oils. Waxes are esters of fatty acids with higher molecular weight monohydric alcohols. Examples include lanolin, beeswax, and whale sperm oil. Complex lipids are esters of fatty acids with alcohol containing additional or prosthetic groups. They are subclassified into phospholipids, glycolipids, and lipoproteins. Phospholipids are composed of fatty acid, alcohol, and phosphoric acid. They frequently have nitrogen-containing bases. Phospholipids may be classified based on the type of alcohol present. Glycerophospholipids have glycerol as the alcohol, including compounds like phosphatidylcholine and cardiolipins. Sphingophospholipids have sphingosine as the alcohol, such as sphingomyelins. This slide shows a visual representation of glycerophospholipids. This slide shows a visual representation of a sphingophospholipid. Phospholipids form structural components of membranes, regulate permeability, and participate in fat absorption and lipid transport through lipoproteins. They prevent fatty liver, serve as precursors for eicosanoids, and are involved in reverse cholesterol transport, blood clotting, and signal transmission. Glycolipids are composed of fatty acid, an alcohol like sphingosine, and a carbohydrate with a nitrogen base, but they do not contain a phosphate group. Examples include cerebrosides and gangliosides. This slide shows the general structure of glycolipids. Lipoproteins are complex lipids with a protein prosthetic group. They include chylomicrons, VLDL, LDL, and HDL. This slide shows the structure and composition of lipoproteins. Derived lipids are obtained from the hydrolysis of simple and complex lipids. Examples include fatty acids, steroids, cholesterol, and vitamins A and D. Lipids function as energy storage, structural components of cell membranes, precursors of hormones, thermal insulators, and protection for internal organs. Lipids also help absorb fat-soluble vitamins, transport lipids as lipoproteins, act as surfactants, improve taste, and serve as electric insulators in neurons. Fatty acids are naturally occurring carboxylic acids with unbranched, usually even-numbered carbon chains. They can be long, medium, or short chain, and are classified as saturated or unsaturated. Saturated fatty acids contain only single carbon-carbon bonds, like palmitic acid. Unsaturated fatty acids contain one or more double bonds, such as oleic acid. This slide shows a diagram of saturated and unsaturated fatty acids. Prostaglandins are derivatives of polyunsaturated fatty acids. There are two essential fatty acids, alpha-linolenic acid, or LNA, an omega-3 fatty acid, and linoleic acid, or LA, an omega-6 fatty acid, which the body cannot manufacture. Essential fatty acids are required for membrane structure and function, cholesterol transport, lipoprotein formation, prevention of fatty liver, and synthesis of eicosanoids. Rancidity is the deterioration of fats and oils resulting in an unpleasant taste caused by exposure to air, moisture, light, or bacteria. It can be hydrolytic or oxidative. The acid number checks purity by measuring the free fatty acids present. A high acid number indicates decomposition and makes oils unsafe for consumption. The saponification number measures the average molecular size of fatty acids. A higher value indicates shorter chain fatty acids. The iodine number indicates the degree of unsaturation. It is directly proportional to the unsaturated fatty acid content and helps detect adulteration. The Reichert-Meisel, or RM, number tests the purity of butter by measuring soluble volatile fatty acids. A typical butter RM number is 25 to 30. Esterification is a chemical reaction of fatty acids with alcohols to form esters in water. Acid hydrolysis is the reverse of esterification, producing fatty acids from esters. Saponification is the base-catalyzed hydrolysis of an ester, producing an alcohol and an ionized salt, which is a soap. Soaps form micelles that dissolve oil and dirt particles. This slide marks the end of the video.